Alright, ladies and gentlemen, welcome back. We're looking at antibiotic resistance and how the body responds to an infection through its immunity. Alright, let's get going here. So first of all, we just want to do a little recap on what antibiotics are and what they do. We talked about this a little bit in the bacteria section when we talked about what some antibiotics like penicillin do in order to break it down. So all an antibiotic is, it's something that kills living cells. Okay? And one thing that you need to realize is that anything with antibiotic resistance, so certain bacteria, they're able to withstand or resist certain antibiotics or sometimes multiple antibiotics. So let's look at the way that antibiotics work. So first of all, some antibiotics are able to inhibit DNA and RNA synthesis, which obviously has a big impact because if they're not synthesizing DNA and RNA, they can't make proteins. So no proteins made. And the reason why that's important is proteins pretty much carry out all of the functions. Okay, you don't have to know the names. These are just some examples for you to look at. Inhibition of protein synthesis. So similar to the DNA, but it's just different mechanisms that it destroys. So this one destroys DNA, RNA being able to synthesize, and then this one is actually the protein synthesis procedure in the ribosome won't be able to happen. So just little things, little differences. Next one, cell membrane breakdown. So we remember in a bacteria we have a cell wall and then also the cell membrane. If the cell membrane isn't functioning properly, it's not allowing things to go in and out, won't be able to function. Inhibiting enzyme action. Enzymes pretty much facilitate all the chemical reactions in the body, reactions in the bacteria. So if those chemical reactions aren't able to be carried out, the bacteria won't be able to survive. And then the last one, cell wall breakdown. The penicillin that we talked about last time, it attacks the peptidoglycan in the walls and causes it to break down. And if you don't have your structure, it won't be able to survive. All right, so let's look at about how some of these bacteria are becoming antibiotic resistant. So this is just our normal bacteria. It has its DNA here. But what can happen is one of three things. The first one we'll look at is conjugation. We've talked about this before. And this is where the plasmid, which is a version of DNA, is transferred from one bacteria to the other. Okay, so that's this one here. So you have your plasmid, it's DNA, it's got resistant gene to a certain antibiotic. It comes, and now since that plasmid's inside the other bacteria, it now can synthesize the gene that has that resistance to an antibiotic. The second one is called transduction. And what transduction is, it's where a virus carries DNA for antibiotic resistance. And brings it to the new bacterium and inserts it. So inserts the DNA into the bacteria. And again, now it can start synthesizing the genes for that antibiotic resistance. And the third one is just if it's called transformation, and this is the last one here, and that's where a bacteria dies, breaks apart, and then it has the resistant gene that gets taken in by the other cell. So resistance gene uptaken, that makes sense, if that's even a word, by bacteria. Okay, so making sure that we know those three. So you might want to pause to get it down. It's pretty messy. Hopefully you can read that. So here's just a little cartoon. How do we actually defend against these pathogens? Fortunately, we have more for our defense mechanisms than just the curtain. So what does it do? So there's two, whoa, my goodness, that is just fast. So we have two big ones here. We have non-specific versus specific. Non-specific just means that it's not going to identify, so it's not specific to a certain pathogen. Okay, so this is just broad. It's looking to protect against any pathogen. So this is our skin, mucous membranes, sweat, oil, ciliated cells, acid in our stomach. So for example, if we ingest some sort of bacterium, hopefully that pH that low pH, that acid in our stomach, will break down that cell, will kill the bacteria. But we also have specific defenses that are a lot more beneficial. I would just write down these 4 cells, and we'll talk about what they do in a second. So you might want to pause it to get it down because we're going to move on. So we have so this foreign body refers to any sort of bacteria, or it can be a virus, or even a protist, pretty much anything that can cause an organism to get sick. It invades the body, and what we have is this macrophage, which is pretty much a big white blood cell. And what it does is it ingests the foreign body, so eats it. And what happens after it eats it, it displays part of that foreign body, so part of that bacteria, which we call an antigen, on its outside so other cells can recognize it.
Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces.
The other cells are things called T helper cells, and they read and recognize these antigens. And what benefit that is these T helper cells send signals to activate another one called B cells. And these ones are really important because what they can do is they produce antibodies. And antibodies target those specific viruses, bacteria, or protists. And once they find the ones they're looking for, they bind to it or they attach.
With a T helper cell that can recognize the same antigen. During the interaction, cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on.
And what happens is when they attach to these foreign bodies, it's pretty much like a homing device, a beacon that sends a signal to a macrophage to say, come find me and destroy whatever I have. And this macrophage comes and destroys the antibodies that are holding on to that foreign body and the foreign body.
The surfaces of the pathogens, marking them for destruction by macrophages.
And then finally, when all of our foreign bodies are gone, a T suppressor cell sends a signal to stop the immune response. What we're looking at next is what a vaccine is. So you guys have all probably had vaccinations or immunizations, and what a vaccine is, it's basically inserting a weakened or dead, or we can say inactive, strain of a certain pathogen. And the whole point of this is to stimulate our immune system using the past immune response that we just talked about. So what happens is you have this foreign body come in, we have our macrophage is going to ingest it just like it did before. It displays its antigen, the T helper cells comes and identifies it, sends the signal to the B cell. The B cell makes some antibiotics, or not antibiotics, some antibodies. And what it does is when you have all these antibodies produced, now if the real thing comes in, it's already going to be ready to go and it's going to attack it really quickly.